Okay, so here shows the steps of meiosis. And this side is going to be meiosis 1, or the reduction division, and over here shows meiosis 2. So we start over here with early prophase 1. There are 4 chromosomes, so the 2N number is going to be 4. There are 2 pairs of homologs right here, and the nucleus is still intact. Next, we go into late prophase, where the homologs get together next to each other and form tetrads, and the nucleus is starting to dissolve a little bit. And then here you can kind of see the spindles start to form, the spindle fibers start to form. Next, we go into metaphase, where the chromosomes align along the midline of the cell. The homologous pairs are next to each other, and here some crossing over happens to create some genetic diversity there. And basically, the chromatin line up on the equator of the cell, and with the help of spindle fibers here that were formed over here. Next, we have anaphase 2, where the homologous pairs separate from each other and move towards the opposite end of the cells, towards the centrioles, which are these structures here. And then after that, we move into telophase, where the cell starts to cleave, and we get 2 cells with formed nuclei, and these 2 cells are haploid because they only have one of the homologous pairs. So next, we go into meiosis 2, which is also called, or known for being, the separation of sister chromatids. So the process is very similar. So we start with prophase 2, where the nucleus starts to dissolve and the spindle fibers form. Next, we go into metaphase 2, where the sister chromatids will line on the midline. Then the sister chromatids split in anaphase 2. And then we have telophase 2, where the cells start to cleave. And last, we end with 4 daughter cells from the original cell up here that are haploid and are genetically different. So here we have the early stages of development. We start off with the secondary oocyte, which has just been ovulated from the ovary. This secondary oocyte moves into the uterine tubes with the help of these fimbriae. And once a sperm cell comes in and is able to fertilize the oocyte, the next stages of development start. So this is right here would be fertilization with a sperm cell and the secondary oocyte in the polar body. Here we have basically the first day of development, with the sperm and the ovum in the polar bodies. This, the ovum and the sperm will unite to form a diploid cell called the zygote, which is shown right here. And then after this, the zygote will go through many stages of mitosis. It will divide into 2 cells, then 4, then 8, and then many more. So these cells are named by how many cells they have. So this is this 2-cell stage, the 4-cell stage, and the 8-cell stage. And usually these 3 stages happen on day 1, day 2, and day 3. And next we have the morula, which is where the zygote usually enters the uterus. All of the previous steps were happening within the uterine tubes. So once it enters the uterus, the morula will develop into a blastocyst, which is this structure here. There's usually a cavity within it, and then it develops into this structure, which is a more developed blastocyte that has an inner cell mass, tryptoblast, and also still has the blastocyst cavity. And this inner cell mass right here is what's going to become the embryo, and then the tryptoblast, which is the outside portion, will become the placenta, and basically the structure that provides nutrients and all that to the embryo here. So usually this will become implanted into the uterus, so we can see it here. It's implanted on the uterine wall. Usually this happens around day 6 to 7 after fertilization.